Shiva, you tell me your chronological age, which is the time passed since you were born. But your body keeps count of time through biological age, which measures damage to cells, and this damage causes aging. How do you think the body coordinates all its myriad processes? It does so by using a biological clock to measure the physical, mental, and behavioral changes that follow a 24-hour cycle. DNA methylation is a useful predictor for measuring the biological age, which indicates your health span, which is the period of time spent in good health. The process occurs due to the covalent transfer of a methyl group to the C5 position of the cytosine ring of DNA in the presence of an enzyme called DNA methyltransferase. It occurs mostly in CpG sites, which are regions of DNA where a cytosine nucleotide is followed by a guanine nucleotide along its 5' to 3' direction. CpG sites are promoters of a gene. Promoters initiate gene transcription by binding proteins such as RNA polymerase and transcription factors. But when methyl groups are attached, this binding is blocked, allowing for stable silencing of a gene as the DNA can no longer be transcripted. DNA methylation can set a gene off. This is why body cells differ based on location and function because different sets of genes are off even though the base genome is the same. Measuring the accumulation of methyl groups on DNA allows us to determine the biological age of a subject, and this is known as the DNA methylation clock. Its advantage is that it allows us to measure cell specific aging, which is more accurate than chronological age in predicting related illnesses such as neurodegenerative diseases like Alzheimer's. Such diseases often share epigenetic patterns, giving us more opportunities of cellular therapy. The various DNAM clocks reflect different aspects of a multidimensional aging process such as telomere biology, epigenomic instability, and senescence. But clocks can be set back, and by understanding what makes the DNAM clock tick, we can possibly reverse aging. One theory suggests that changes in the amount of stem cells throughout life initiates the process, while the other theory suggests that damage to DNA induces CGP methylation when breakages are repaired. One cool company that is researching this is Base Genomics, and recently they have raised $11 million to develop an enzyme to possibly cure aging. But until a cure to aging is found, you can manage the expression of your genes by paying attention to your diet and physical activity to take care of your biological health. So, until then, stay curious, stay healthy.